So we've talked about ingestion, we've talked about digestion. Now we're moving on to the third phase of the digestive process in humans, which is absorption. And absorption of different kinds take place in the small and the large intestine. And so here's where we are. Let's just take note of where we are on our journey. We've looked at our food being mechanically digested through churning, and also chemically digested via hydrochloric acid and some important enzymes. But when food is ready to move from the low pH environment of the stomach into the intestine, there are some things that we need to be aware of. So let me just remind you of a few key ideas. So first of all, in our last video, we talked about how pepsin, which is present in the stomach, breaks down proteins into polypeptides, right? And that pepsin what is the on switch for pepsin is that low pH. The thing about the stomach, though, is that, it has lots of mucus that's there to protect the stomach from digesting itself with those acids, right? And also, the muscles in the stomach are all protein, protein, and so we don't want pepsin to digest the stomach itself. So, that's why the mucus is there to help with all of that. But the problem is that the same amount of mucus we don't see formed in the small intestine. So there needs to be a way to switch pepsin off so that it ceases to break down, larger protein molecules like the muscles in the stomach. So this is how it happens. So the mucus in the stomach wall prevents that, that digestion from happening from pepsin just eating away at the stomach. However, there is a drastic pH change once food moves from the stomach to the small intestine. It's not shown in here, but we have these digestive accessory organs like our liver, gallbladder, and our pancreas, and they're injecting the first section of our small intestine with some digestive juices that are going to change the pH of and change the pH of our small intestine. So what that does is that is the off switch for pepsin, right? That shocking high rise in pH is going to inactivate our pepsin. It's going to turn it off. But it's going to that higher pH level is also going to be an on switch for another important enzyme, and that is trypsin. And it's really too bad because if we had been in class, we would have done a super cool lab using boiled eggs and pepsin and trypsin, and we would have seen how these pH differences really do affect the breakdown of proteins, right? So remember, pepsin is breaking down large proteins into smaller polypeptides. Trypsin has a job. It can't break down large proteins into polypeptides. It can only take smaller polypeptides and make them into amino acids. So it's not going to digest the intestine, not normally, right? So it's okay for trypsin to be there in that small intestine because it doesn't have the ability to break down these thick muscular walls that we see in our small intestine. Okay, next. So let's talk a little bit more about the small intestine. We're going to look at, first of all, what its overall job is, but then we're going to break it down into different sort of subsections. In grade 10, you would have learned that the small intestine's job ultimately is to take in nutrients and absorb them into the body's circulatory system so that they can be brought to cells and then used for cellular respiration, right? That's how the digestive and circulatory systems kind of work together. They help each other out. The small intestine, this year we're going to divide into three sections, each with sort of more particularities to them. So let's look at this diagram. Now here we can see the stomach, and in yellow, purple, and pinkish, we can see the three subsections of the small intestine. So the first subsection of the small intestine is the duodenum. Okay, and I'm hoping already that you can see that it seems to be quite a bit shorter than the other sections. So food will move from the stomach to the duodenum. Then it will migrate, be moved through the jejunum. And the jejunum is quite a bit longer. It's the longest section of the small intestine. And then from there, food will move into the ileum. Okay, so each of these different structural components of the small intestine has its own functionality and sort of anatomical features, structural features that we're going to discuss today. So here we are focusing on the duodenum. So first of all, the duodenum is only about 25 centimeters long, and we're going to do a recap of at the end here to kind of think about just how long our small and large intestines are together, all wrapped up in that small little space in our abdominal cavity. It's pretty amazing, really. So it is U-shaped, and that U-shaped is, here we have the J-shaped stomach, but the U-shaped, sort of almost C-shaped really, duodenum. So that's what it looks like, but let's kind of dig a little bit more deeper. And you would have seen this, I think, last year. I'm going to pull this all up because I think that the pictures are helpful here. So all along the interior of our duodenum and our jejunum and our ileum, so the entire, small intestine to varying degrees, what you're going to see are these little kind of hills, if you will, which are called villi. So we would have a single villus and multiple villi, if we're looking at plural versus singular. And what they serve to do, you can think about it almost like if you had a piece of paper that was scrunched up into a small ball. That small ball still has the same surface area of paper, it's just squished into a smaller area. So by having all of these villi together, what we do is we vastly increase the surface area through which our nutrients, our small little broken down nutrients, can be absorbed. So here's our cross section of any part of the small intestine, and you'll see those villi. But I also want you to note, here's a second picture. So here you can see a villus. Here's another villus. But on each villus, there are little mini villi. Those are called microvilli. And again, that just serves to enhance the surface area for absorption of nutrients. Now, we're going to talk about something else. I'm hoping that you'll notice that each of these villi has its own little blood capillary system, right? So capillaries are really fine blood vessels, so that attach our small intestine to the rest of our circulatory system. So that when, let's say I have here's a little, glucose molecule that's been broken down from a starch, and it gets close to the villus, and then it's going to be able to absorb through the wall of the villus and right directly into the circulatory system. That's kind of how that works. Let's write a little bit down about that. So the two features, that being the villi and the microvilli, are going to vastly enhance or increase the surface area so that you can optimize the absorption of nutrients that are moving through there, digestive products of the breakdown of our food. And each of those villi is going to be supplied with a network of capillaries. I forgot to mention lymph vessels as well, that are just going to support the whole absorption process. Okay, that's essentially what's going on there. So next, what we need to talk about is the fact that the duodenum is where, remember I mentioned earlier that the gallbladder and the pancreas are kind of those accessory organs right close to the duodenum, and they're actually connected to the duodenum and are injecting it with some pretty important digestive enzymes and such. So bile, for instance, and some other digestive materials, insulin, but also some pancreatic amylase and so on and so forth. So let's just take a look at this to kind of get a sense of what we're talking about. I really liked this diagram because it zooms in on all the important things I want to be talking about. Here is our duodenum, right here. And the duodenum is adjacent to both the pancreas and the liver. And then there's also the gallbladder just kind of tucked under the liver. And so it's not easy to see right here, but I'm going to do my best, and then I'm going to pull up a different diagram. But you can see how the gallbladder and the liver are connected through this little duct here. And then that duct kind of comes together as a fork in the road, if you will. And that's where the bile coming from being made in the liver, stored in the gallbladder, can be injected into the duodenum right there. Likewise, the pancreas, is going to be injecting the duodenum with some of its digestive enzymes and such. And it does so by way of this pancreatic duct right here. And so the injection of those fluids is going to, again, you'll remember that over here we have the stomach with its low pH. The injection of these fluids into the duodenum from the gallbladder, liver, and pancreas are going to raise the pH of the duodenum really quickly and inactivate pepsin and then activate trypsin. Here's just a zoomed in look of the duodenum with the ducts that I just spoke about. So the liver, it would be up here. Here's from the liver. There's the gallbladder duct, and then the pancreatic duct, which both kind of come together and inject into the duodenum. Well, that is that. So again, this may be a diagram that you will want to have handy for you as well as you're studying for our digestion quiz. So we've talked about the duodenum. Next, we're going to move on to the jejunum, and we've done most of the discussing of the large intestine, or sorry, small intestine as a whole when we talk about the duodenum, because the jejunum and the ileum, they still have those microvilli that are lined with the capillaries and lymph vessels, right? That is there as well. It's just that they're not so closely proximal to our liver, gallbladder, and our pancreas. So I'm going to talk about how the jejunum and the ileum are just a little bit different than the duodenum. And the first thing that I hope that you're noticing is that it's quite a bit longer than the duodenum. In fact, about 2.5 meters long. So that constitutes about 40% of the small intestine. And sorry, I might have misspoken earlier when I said that the jejunum was longer. The ileum is longer. But we will get to that. So already we're looking at, 25 centimeters for our duodenum, 2.5 meters for our jejunum, right? Think about a meter stick and how long a meter stick is. So kind of take your hands and figure out what that looks like, and then multiply that by 2.5. And that's how long just your jejunum is. And we haven't even talked about the ileum yet. So basically, the jejunum is going to have more folds than the duodenum, just to maximize, absorption. It gives the food more time to move through it and for those nutrients to be absorbed. And it's going to break down any remaining carbs and proteins so that they are small enough, those macromolecules are small enough to pass through the wall of that section of the small intestine. We're going to talk about the ilium last. The ilium is about 3 meters long. So let's do some math. 3 plus 2.5 plus 0.25. You can add that up. That is long. We're looking at almost 6 meters. 6 meter sticks, head to tail, all squeezing into our small little abdominal area. It's pretty substantial. So you're maybe wondering about the math here. How am I getting 60 plus 40? And then I haven't even talked about the duodenum yet. We're going to round to the nearest 10 here and just, think about it as sort of just around the right values. You could probably dig deeper and find the actual percentages, but that's just to give us a roundabout idea. Now, you're going to see fewer villi and microvilli in our ileum as compared to the duodenum and the jejunum. And the reason is for that, perhaps we've evolved that way. But at this point, we've had our nutrients moving through our system a fair distance, right? And so the need for as great a surface area may not be there at this point. But we're going to still see some continued absorption through the walls of the ileum. Okay, now at the end of the ileum, and I'm going to show you a little diagram here. At the end of the ileum, what we're going to see is we're going to see food being pushed into the large intestine. So by the time our food moves into our large intestine, any of the nutrients that should have been absorbed ought to have been absorbed into our circulatory system at this point. Okay, so here is a picture of our large intestine. And so just to be clear about this, the large intestine is made up of two components, the colon, okay, which is made up of the ascending means going up, so ascending colon. So the ascending colon would be connected to the ileum, like right about here. And then our food would move up, and then transverse sort of means across. So our transverse colon, and then our descending colon. Now the sigmoid, a sigmoid structure I think means kind of like curvy or S-shaped. So you can sort of see how it has to kind of curve around to reach the rectum, and then eventually the anus, which is also a sphincter. Now we're not going to talk about egestion or elimination because I think it's fairly straightforward. There's not a lot to talk about. The anus is a sphincter, and that's about all there is really to talk about at this level about that. So moving on, we're going to talk about the colon. The colon itself is about 1.5 meters long. So we just finished talking about how the small intestine was just a little bit short of 6 meters long. So why are we calling the large intestine large if it's shorter? So this is why. The large intestine, we talk about large and small, we're talking about basically the circumference of that particular tube, right? So if we go back, let me see if I can quickly get back here. If we look at the circumference of the small intestine, it is quite a bit smaller relative to the large intestine. And we can see that if we look at a comparative picture here. The rectum is about 15 centimeters long. It's quite short relative to the colon. And so let's talk about the particular jobs that our colon and our rectum have in terms of our digestive process. So the function of the colon is the absorption of ... Let's see if you remember from grade 10. Water, mostly water. If you have diarrhea, it's because there's likely to be a problem with possibly the bacterial culture or the environment in your colon, and so water is not getting absorbed and basically flushes through your body. Constipation, on the other hand, involves overabsorption of water, and that usually happens when you're dehydrated or your body doesn't have enough fiber to move your fecal matter through your colon fast enough, and so water just kind of keeps getting absorbed and absorbed and absorbed, right? So if you're constipated, definitely increase your fiber content. Some other things that are absorbed are some salts, right? That often has to do with maintaining osmotic pressure in your blood vessels, as well as some other vitamins and minerals. Right, so you may remember early on in the year when we were talking about bacteria and how bacteria are not always bad, and also how E. coli bacteria is not always bad. Our colon is like this microbiome. It's like its own little world where bacteria grow, thrive, and have little jobs to do. So E. coli will live in that large intestine. They're going to take that waste and use it for their own purposes, but it's a symbiotic relationship in that, those E. coli that are hard at work are busy making us some important vitamins B and K. So we need them. They're really important for us. So lastly, I just want to talk a little bit about some questions that students usually have at this point. So just to talk briefly about the rectum being about 15 centimeters long. Its job primarily is to store feces in preparation for being eliminated. That's pretty much it. I haven't included in this note, but you may want to add that to your notes. So students will typically ask me a few questions. If we were in class, we would get to this, so I'm just going to kind of guess that this might be on your mind. So students say, well, how long does all this take, right? So when I swallow a piece of hamburger to the point where it is making its way into a toilet bowl, how long does that take? Luckily, your textbook has this lovely little chart that outlines the length, approximate length, and of course, this is very subject to, people have different metabolisms, different metabolic rates. So, often it'll give us a range, and the range is quite high. So, the primary function of the mouth we know is for that initial mechanical chewing and chemical enzyme digestion. Um, and that usually takes between 5 and 30 seconds. But better to be in the 30-second zone because that's just better overall for your digestive health, allows your body to break down your food more into smaller pieces, and also to give your stomach time to sense fullness. So there we go. The esophagus, and I don't know if you've ever felt it, but if you swallowed something that you didn't chew enough, you can definitely feel it going down your esophagus. Likewise, you also can typically feel something very, very hot moving down your esophagus, and that is not very comfortable, but that whole process takes about 10 seconds. Now, stomach. This is like waiting for the cable guy. They say, oh, I'll be there between like 8 a.m. and 5 p.m. So, your food can be, depending on the type of food and what it is that you've eaten, that process of breaking down food in the stomach can be anywhere from 2 to 24 hours. And again, that often has to do with factors like metabolic rate. Same with the small intestine. So about 3 to 4 hours for food to move through by peristalsis through your small intestine where nutrients get absorbed, and then eventually into your large intestine where the primary function is water absorption, and that takes like anywhere from a day and a half to 2 days typically. All right, next question. Students typically ask, why is feces brown? What gives it that brown color? Well, this is the short answer, and the long answer you can Google it. But the brown fecal color that we sort of anticipate in our excrement is due to this compound called bilirubin. Now, bilirubin is basically a byproduct of the breakdown of hemoglobin. So remember, hemoglobin is that really important oxygen carrying molecule that is a part of our blood. And so, our body's going to be using and then reproducing red blood cells, but some of those red blood cells are gonna be broken down. And so that's what that is. The brown color is a breakdown of our hemoglobin. And the molecule specifically, bilirubin. Now, if you wanted to, and I'm not going to indulge us by going through this, but it's like this, never-ending rabbit hole of, what would you call them? Diagrams, pamphlets about, the color of your poop, right? It's endless. If you Google this, there's lots of information about how the color of your fecal matter can give you an indication potentially about illnesses you might have. Your typical fecal matter is going to be brown. If it's any other color than that, maybe see your doctor. I'm not a doctor, but maybe see a doctor. That's all I have to say about that. And then, that, discussion around the color of feces usually brings us to why it smells badly. And so I'm going to remind you that in our intestines, we have those E. coli bacteria, to mention one. Now, those bacteria are going to be busy eating away at some of the waste materials that we haven't used as nutrients. And so the byproduct of those bacteria digesting our waste is going to be gases, and those gases often don't smell very good. So that's where that fecal odor comes from, but it is absolutely normal in most cases. Some people will also ask at this point, well, why is it that some people have fecal matter that smells different than other people's fecal matter? That can be a function of a lot of things, like A, what food you're eating, and also B, the type of bacterial cultures that are present in that microbiome that is your large intestine. So I hope that you were able to make sense of this longer last screencast on absorption. And of course, as always, if you have any questions, make sure that you stop by our Zoom meeting that will be live all day on Wednesday to ask questions or to clarify or just to say hello.